And this is what we see when we roll all those data up into a summary. We see this first plot is the amount of material at biomass. It's just the amount of periphyton we have out there in red in the Everglades. And then these green bars are all these different kinds of ecosystems from all over the world, just a summary from what's been published in the literature. And so you see that there's tons of periphyton relative to every other wetland out there. When we look at the amount of phosphorus that's actually in the algae, we see that it's really low, as we would expect with not very much being in the water out there. And so we can ask, this is a paradox. It's weird. Is it unique? Is it novel? Is it distinctive? In order to answer that, we started going all around the world and looking at these kinds of environments that have that limestone underneath them. And we were able to sample in the Yucatan Peninsula of Mexico and in Belize and in Jamaica and even around the Great Lakes, which might come as a surprise. But there's tons of little wetlands all around the North Shore, particularly of Lake Huron and up in the Bruce Peninsula of Lake Huron, that are just full of these little wetlands that are on limestone, and for all the world, they look like the Everglades. Big surprise, way up in the temperate zone. And so this is our field crew from our work down in Mexico. We stayed at these lovely places and got to sample in these incredible marshes and had a really wonderful team from the University of Mexico that really helped us and worked closely with us, particularly on the animal part of what we were doing. And this is what we found. So in each of these places, we were sampling the same stuff, using those different kinds of traps and picking up the algae from the water and the little animals that live in it. And in every place, we found tons of this kind of calcareous, with lots of limestone and calcium carbonate in it, periphyton mats in every place. And so they highly resemble, just from the get-go, the Everglades. And we started looking at the history of use of these landscapes and the history of what they might have looked like in the past to think about this question of how much have these systems changed. And in fact, it looked like at least some studies had shown that periphyton was used as fertilizer. So it was taking what nutrients were in the water column out, because the little algae are good at that. And then the Mayans would harvest it and move it clear over to some other part of the landscape that they were living on and use it for like as an organic fertilizer. So kind of a cool idea there. Even up in Canada, these are the little sites in Canada. This is Utricularia foliosa, which is in one of these pictures here. I know Pauline Goldsmith painted a beautiful one. Somewhere in here is it. The purple bladder, or the folious bladderwort that lives all over the Everglades. You know, here it is. This is Cladium. This is Eleagris, different species than we have here, but the same genus of plants that live, same genera that live in the Everglades. And you can see all the periphyton there. And this is just looking out across one of these. They call them alvar wetlands that looks out into the Great Lakes. So when we put these other ecosystems on the same chart, so Everglades in red, all the other wetlands of the world in green, we see that these other wetlands also have lots and lots more periphyton than any of these other wetlands around. And they have very, very little amounts of nutrients available to them. So when we think about this, you know, is it unique? Well, in terms of this periphyton attribute, not really. And I'll show you a little bit more about the composition of these communities to reinforce that. It's not. There are other habitats out there quite a bit like this. Is it novel? Well, these communities have been around for some time, and our historical data and our paleohistorical data are showing us that. But it seems certainly distinctive compared to all these other wetlands in the world. We have here a global representative that's globally known, globally acknowledged to be super cool, being sort of an advocate for these other wetlands of the world. That's kind of the way I like to think of it. And so when we think about this idea of the Everglades just being full of this periphyton, I love this kind of little bug's eye view looking through the floating paraphyton mat. If you take a little sample of that and you put it under the microscope, and that's what we spend a lot of time doing in my lab, you find these stringy things like this. These are blue-green algae that make up the kind of backbone of it. Lots of calcium carbonate crystals, all those little stringers hanging on to this blue-green algal filament are calcium carbonate. And lots of, this is a diatom with lots of jelly, kind of, that it puts this jelly out into the environment to hold itself in the mat. And all of those things, sorry, I keep hitting the wrong button. All those things are really important to getting nutrients and keeping them. So these communities have figured out how to deal with having hardly any nutrients available to them. They take whatever it is that's out there really efficiently. They keep it, they trade it. These little bacteria are trading it with the algae all the time. And through all this enzyme stuff that they do. All this stringy stuff, this mucilage, it's snotty stuff inside the paraphyton is really good at holding on to nutrients. So they have it figured out. So if we look closely at what's in there, so what about the species that occupy this thing? So a little magnifying glass, look at it, the diatoms that we find in there. Here are the different species. These are diatoms. These are the glass-encased, beautiful algae that are all over the world. One of the most dominant groups of algae in the world, produces 1 out of every 3 molecules of oxygen that we breathe, so an important organism on the planet. And super diverse. Anywhere you go around here, you're going to find diatoms. And these are the species that we find in the Everglades, and the ones with the low red dots are ones that we had never, before working down here, seen in other places before. And I have to point out that Nancy Maynard, I'm going to get you with the beacon here, but was the first person to really work on these mats and discover some of the species that are growing in there. So you're probably recognizing some friends there. So we indeed think about these things as our friends because they are not things that are found anywhere else in North America. I've studied diatoms all my life, never saw them before. And if we look at how many species there are in each sample, we find in lots of other wetlands, usually in one slide you find 40, 50 species, a lot of them. In the Everglades, we only find them. It's like a family of ones that are always living together, and that's who you see. And so again, is this paradox unique, novel, distinctive? We will turn to our sites in the Caribbean and in Canada, and lo and behold, the same ones. And so this was a huge surprise. It's like finding a family member in another country. Things that you only ever saw in the Everglades, and you go somewhere else, and there they are. And wow, this was totally fun for all of us. We put a new name on this species. Actually, I haven't updated the slide. This actually is endemic to down here. And we indeed see that these other communities all around the world in these other kinds of, or in the Caribbean and Canada and these other kinds of similar habitats have really low diversity as well. And so that's a distinctive feature. Not novel particularly, I'll show you why, not unique, but certainly distinctive. Are we losing this distinctiveness with this kind of enrichment we've allowed to happen through the canals that we put in the drainage of that water into the Everglades? Well, we've experimented around with this. David Lee was a big part of this project where we set up an experiment to put a whole bunch of nutrients into the Everglades to figure out what is that level at which we have to protect the system. And so this is looking down on a little square meter plot, and we put 30 parts per billion in. Remember, the background is at 10, so that's like triple the background. And the paraphyton went away, completely disappeared. Another paradox, completely the opposite of anything you would expect a plant to do when you give it more nutrients. And so if we look at these same plots again, here's all the other wetlands, Everglades, the other places that are like the Everglades, and then the Everglades plus phosphorus. We send the phosphorus values up in those mats to an extreme level, and we get rid of the floating calcareous mat. It disappears. Major, major change in the system. And we also change the species that are there. And we bring out ones, none of these ones with the little pink dot, those are the ones that show up with the addition of phosphorus. None of those are dominant in the natural part of the system. None of them are endemic to the Everglades. None of them are things that we only find here and in the Caribbean. Instead, they're things that live all around the world. I mean, this was one of the first diagrams I saw when I was working out in Iowa. This lives all over the world in very enriched habitats. So what we're doing by putting nutrients into the ecosystem is taking a system with those distinctive properties and turning it into one with novel properties, with properties that are novel to it, right? With the definition that I threw up there. Novel in terms of the ways in which it relates to the history of what it has been. And so we're really re-engineering the system when we add nutrients to it. We're losing that periphyte mat. We're changing the little species that live within it. So can we preserve this distinctiveness of the periphyte and all the things that are dependent on it? One of the things that we're trying to do in the system is put in these areas called stormwater treatment areas. There's a bunch of them functioning already where we run the water into them, and the periphyton and the plants and the other organisms that live in it pull the nutrients out. And so you start out with lots of nutrients, and it gets less and less as you put them through the plants, and then the periphyton does this kind of final scouring because it doesn't like a lot of nutrients, so you've got to give it just a little, and it'll pull those out, and then put the water into the Everglades. We need lots more land to do this. Sadly, the South Florida Water Management District just advised our governor to not allocate 40,000 acres of land, which is really desperately needed hectares of land, 40,000. That would have been a huge, huge, huge boost. Our major problem with restoring the Everglades is, you know, we have ways of moving more water south, but that water has to be clean. And this is how we get it clean. And we need lots more acreage of this. And we have the opportunity to do that as a state, and everybody voted in support of Amendment 1, and we need to do it. And hopefully this is not a lost cause. A self-help program for periphyton. I like thinking of it that way. So periphyton helps itself downstream by cleaning up the water.